How do space biology experiments study anemia? Have you ever wondered how astronauts maintain their health in the unique environment of space? One area of focus is anemia, a condition that can affect their performance and well-being. Let's take a closer look at how space biology experiments study anemia aboard the International Space Station. Anemia occurs when the body does not have enough red blood cells to carry oxygen. In space, this condition is often referred to as space anemia. It primarily results from the increased destruction of red blood cells, a process known as hemolysis. In normal conditions on Earth, the body produces and destroys about 2 million red blood cells every second. However, in microgravity, this destruction rate can increase to about 3 million per second. To study this phenomenon, astronauts on the International Space Station participate in biological sampling. This includes regular blood draws and the collection of exhaled breath samples. Researchers analyze these samples to measure biomarkers related to red blood cell destruction. For instance, elevated levels of carbon monoxide in exhaled air indicate increased breakdown of hemoglobin, a key component of red blood cells. Blood tests also play a crucial role in this research. Scientists measure iron levels and iron-binding proteins like transferrin and ferritin. These proteins increase as red blood cells break down and releases their contents. By monitoring these indicators before, during, and after space missions, researchers can track changes over time and better understand the effects of microgravity on red blood cells. Findings from these experiments reveal that red blood cell destruction increases significantly during spaceflight. Studies show a rise of around 54% compared to normal conditions on Earth. This hemolysis begins early in the mission and continues throughout, indicating it is not just a temporary adjustment to the fluid shifts in space. Understanding space anemia is essential for astronaut training and long-duration missions. Pre-flight screening can help identify astronauts who may be at higher risk for anemia. Continuous monitoring during missions ensures that health and performance are managed effectively. Anemia can impact oxygen delivery, muscle strength, and overall endurance, making this research vital for astronaut safety. Moreover, the knowledge gained from studying space anemia could have applications for medical conditions on Earth that involve anemia and muscle or bone loss due to prolonged immobility. By understanding how microgravity affects red blood cells, scientists can develop targeted interventions to protect astronaut health and potentially improve treatments for patients on Earth. Space biology experiments studying anemia provide critical information about how microgravity impacts red blood cell production and destruction. This research not only helps ensure the health of astronauts but also contributes to our understanding of anemia in broader medical contexts.